And it's a great pleasure for me to introduce Dr. Jean Fan from Johns Hopkins. Jean is a computational biologist. So she got her bachelor's at Hopkins from biomedical engineering and applied math. Then she went to do her PhD in bioinformatics and integrative genomics at Harvard. And then she did a postdoc, a brief stint of a postdoc, and then Hopkins recruited her back, and then I guess they're lucky to have you there. So John is in the Department of Computational Biology, and she's won several awards, including the NSF Graduate Research Fellowship, starting from the beginning, and recently the K99 R00. Is that, am I saying that right? Looking at
yeah, I think yeah, there was an F99 K00. There's a couple of these.
Okay, I got confused. Sorry. So an F99, then a K00 mechanism, looking at tumor heterogeneity at the single cell level. So she does amazing work. And I was reading through her papers, and she's done cool work on RNA velocities and how to map them and analyze them. And hopefully she's going to talk about that work today as well. So let's welcome Jean. Her talk is going to be about computational methods for molecular and spatial transcriptomic characterization in the brain.
Great. Well, perfect. Well, thanks so much to Shantanu for the introduction and coordinating this invitation. So today I would like to teach you all a little bit some of the computational methods that we've been developing in my lab, and hopefully inspiring you to think about how to apply these methods and technologies to enhance our understanding of the function and the diseases that impact the brain. Before we get started, I do want you all to note that I am in full screen mode. So I actually cannot see any of your wonderful faces. And I also cannot see if you are raising your hand or anything like that. So if you do have questions, please do hold them to the end, and hopefully I will be able to see them better then. So then let's see. Great. Before we get started, I would just like to highlight some of the friendly faces behind the work that you're about to see. We are a rather new team at Johns Hopkins University. I started my lab in the midst of the pandemic in July, so none of my students have actually had the chance to meet each other, but we're all working remotely and very excited to work together, and we would love to collaborate with you if you are interested in reaching out and starting a conversation. So for my team, like many at the UCLA Brain Mapping Center, we're very interested in understanding, what are the mechanisms behind the brain's various functions. And in order to do that, we believe we really need a type of systematic assessment of the underlying cell types, their spatial organization, their connectivity, and other functional properties. All this is to say is we want to ask questions such as, what are the different cell types in the brain, and how are these cell types spatially organized? Are they connected in any particular way or interacting in any particular way? And how does this spatial organization relate to function? And then ultimately, how are these various features of cell state and spatial organization different in health versus disease? Okay. And so in order to begin addressing these questions, we can take the complex tissues such as the brain, and we can begin measuring at a quantitative and molecular level, what are the genes, let's say, that are being expressed in the healthy brain versus the diseased brain. And perhaps, on average, using techniques such as bulk RNA sequencing and other bulk transcriptomic profiling technologies, we can measure how, on average, across all the different cell types of the brain, certain genes may be upregulated in health and other genes downregulated in healthy brains relative to diseased brains. But these bulk measurements ultimately are going to represent a sort of average of all the cell types in the brain, and the actual perturbations may affect, for example, only a specific cell type or only a specific proportion of a cell type. So then in order to get at that resolution of information, Other techniques such as, let's say, prior sorting, we can sort out our inhibitory neurons or excitatory neurons and do bulk RNA sequencing on the sorted samples. But that still hides a lot of potential additional heterogeneity within these cell types. And then we're further also limited based on what markers we can sort for. So then more recently, single cell transcriptomics has really allowed us to begin profiling on a very high throughput level, what are the genes that are expressed in individual cells. So here is a T-stochastic neighbor embedding representation where each point is a different cell. And we've measured the full transcriptomes for these cells, but we can try to represent it in a low dimensional embedding here and color the cells based on their putative cell types. So cells are closer to each other if they're transcriptionally more similar. And we can begin seeing clusters of inhibitory neurons or clusters of excitatory neurons, oligodendrocyte progenitor cells, and so forth. But still, these single cell transcriptomic technologies currently require dissociation. So we lose out on the spatial organization of these cell types. And, even though we're able to see, for example, that, this transcriptionally distinct inhibitory neuronal subtype is different from this one, it's not clear to us how this may be, how these different cell types may be spatially organized and how that spatial organization may ultimately influence, for example, what genes are being expressed by these cells and so forth. So that's where recent technologies in spatially resolved transcriptomics are now allowing us to not only profile, what genes are being expressed in these cells, but to preserve how they're organized within the tissue. So this is a MERFISH image, which is one type of spatially resolved transcriptomic technology that is able to measure what genes are expressed in individual cells. So each point here is a different cell. And this is the 2D representation here is the actual spatial organization within the mouse preoptic region of the hypothalamus. And just visually, you can begin appreciating how these different cells are being colored based on their different cell types that we've inferred from based on what genes that they express. And visually, we can just see, there are different cell types lining the third ventricle. There are certain cell types that have these different spatial patterns. And we can then start asking certain questions associated with the spatial organization of these different cell types and how that relates to function. And as I mentioned, there are a lot of advances being made in imaging and sequencing that have enabled these spatially resolved transcriptomic profiling measurements. Just a few of the different technologies are highlighted here. This is actually an older slide, so I think now there are actually even more technologies like DividSeq from Rafan's lab has been highlighted here, but there are many others. But one of these techniques, which was pioneered in Xiaowei Zhuang's lab at Harvard, where I conducted my postdoctoral research, it's called multiplexed error robust fluorescence in situ hybridization, or MERFISH. And what MERFISH does at a very upper level or broad level is it enables simultaneous transcriptome measurements in a spatially resolved manner with molecular precision. So it uses, just very, very briefly, it uses combinatorial labeling and error-robust barcoding to represent targeted RNA species as these error-robust barcodes that we can then imprint onto the RNAs using these oligonucleotide oligonucleotide probes that we can then read out with sequential rounds of SMFISH, essentially. And what this means is that after many rounds of imaging, we can decode exactly, which RNAs are, in what spatial organization within fixed cells that are then, within tissues. So for more details regarding MERFISH, I would encourage you to check out the original science publication in 2015. But just overall, at the end of the day, the type of decoded data we get is, for example, here we have a cell. The cell has lots of different RNAs, but we can image sequentially, a targeted set. And after many rounds of imaging, we can decode like this spot here to correspond to gene A, and likewise this and this and this. And we can do this, of course, for a targeted set of genes not only for 1 or 2 genes, but now for hundreds to even up to 10,000 genes. And of course, we can do this not only for a single fixed cell, but we can do this for hundreds and to millions of cells within fixed cultures and within also tissues. And of course, we can, in addition to appreciating the type of molecular organization of these RNAs within the cell, we can, for example, segment the images and get these regions that are associated with one cell and begin counting, how many of each different RNA species is within the cell. And that allows us to achieve spatially resolved single cell resolution transcriptomic measurements. And then, we can represent this data, for example, as, for each cell, how many copies of each gene do we observe? And of course, we have millions of cells and up to 10,000 genes now. So this is, as you can just visually appreciate, it's very large data. And of course, it will take a very long time if we were to just manually look through these genes one by one. So we really began needing computational methods just to be able to grapple with this data, and also statistical approaches to be able to begin trying to recover biological insights. So to learn more about some of the single cell and spatial transcriptomic analysis methods, I would encourage you to check out our reasonably recent review in Nature EMM, where we discuss some of the computational approaches that are being taken for analyzing this data. But today, I just wanted to first highlight, with this technology like MERFISH, and how can we combine it with interesting computational analysis methods? What questions can we address? So recently, this is work led by Chenlong Xia. During my postdoc, we collaborated on a project where we sought to combine MERFISH with expansion microscopy to be able to measure 10,000 genes within fixed cultured U2OS cells. So this is cultured cells within a dish, but it hopefully serves as a proof of concept to how we can then apply these to other cell types and ultimately within tissues. But we were able to apply MERFISH to image and detect 10,000 different gene species with a very high detection efficiency and a very low misidentification rate. And given this data, we can now begin asking some interesting questions, such as what, for example, we can look at an intracellular level. What is the spatial organization of RNAs within the cell? Can we identify different genes that are, for example, enriched in different subcellular compartments? One subcellular compartment we looked into was the endoplasmic reticulum, which you can see here. It's the background. That's an image where the white corresponds to the endoplasmic reticulum, and each dot here is a different mRNA. So we can ask, for example, statistically speaking, are there certain RNAs that appear enriched within the white pixels and depleted from the black pixels that are not the endoplasmic reticulum? And indeed, we can identify a large number of genes that we can then visualize and overlay onto the actual ER staining image and just begin appreciating visually how indeed the RNAs that we've statistically identified as being enriched are actually, lining up very nicely with even the very intricate networks of the rough ER. And likewise, so that's, we can begin asking questions on a intracellular level, but we can also zoom a bit more outward and look on the cellular level and begin asking questions related to, related to the subcellular organization of, in this case, different cell states within the culture dish. So again, here you have each point is a single cell, and they've been colored based on their transcriptional state in this case. But the XY coordinate is actually their spatial organization within the culture, tissue culture. And just visually, you can begin appreciating how certain cells at certain cell states tend to be spatially co-localized with each other. And we can also test on a more statistical quantitative level if we look within the k nearest neighbors in space of, for example, all the yellow cells, what is the distribution of cell states that we observe? And indeed, quantitatively, we can see that if you're in the yellow state, you're more likely to be spatially adjacent to other cells in a similar transcriptional state. And for just, I'm going over this very briefly, but for more details, please do check out our paper in PNAS. And I think for this crowd, perhaps more excitingly, we've applied these techniques to U2OS cells, but of course we can apply them to other cell types as well. So Guiping Wang, who is a graduate student in Xiaowei's lab, who's now graduated and is currently pursuing her postdoc in Howard Chan's group at Stanford, not too far from you all. She led an effort to use MERFISH to characterize the spatial organization of the transcriptome in individual neurons. So this was where she applied MERFISH to characterize the spatial organization of different RNAs within cultured hippocampal neurons, actually. And specifically, she was able to measure up to 4,000 different RNA species and began computationally analyzing, for example, what are the different RNA species that appear to be co-localized to the soma, to the dendrite and to the axon, and do we see different patterns of subdendritic distributions of RNAs? And I think having these molecular imaging-based approaches, we can actually you can see here how we have these individual neurons, and there are different RNAs. Each point here is a different RNA, and they're colored based on their species. And there are just certain RNAs that are going to be co-localized to different polarized compartments within the neuron. And so for more details regarding this paper, please do check out her preprint, which is available on bioRxiv. And as you can imagine, even just visualizing this data can become a little bit challenging. It's hard sometimes to, let's say, visualize all the different gene species and to make a lot of different plots, one for each gene species. So we actually developed an interactive browser to be able to just interactively visualize and play with this type of data. So what you see here is, again, this is the neuron MERFISH data that I just mentioned, where each in the background is actually a tau map2 staining of the neurons, but each point is a different mRNA that we're detecting. Rather than showing all the mRNAs, I've only highlighted a few here. And just visually, you can begin appreciating, for example, it's like, oh, there are certain inhibitory neurons marked by GAD2, and there's excitatory neurons marked by SLC17A7. And we have, in red, CAMK2A, which localizes a bit more to the distal dendrites and less in the soma. And just visually began appreciating not only the transcriptional heterogeneity of different cell types, but also the spatial organization of these RNAs within the neurons themselves. But the thing I really want to focus on today, as Shantanu mentioned, is that one major limitation of all of these single cell and spatially resolved profiling technologies right now, at least, is that these cells are fixed, so they're dead. There's no way for us to, for example, follow the cell as they mature, as they divide, and see what are the different genes that become upregulated and downregulated as these cells go through these different dynamic changes. Unfortunately, at least as of current, these expression measurements represent these static snapshots in time. And I would argue that this is, similar to, a picture here that we have here of a group of friends, jumping off a cliff. That, we see this as a single static snapshot in time, But of course, we're smart enough that even though this is a single static snapshot, we can essentially infer a certain level of dynamic information. And even though this is a static snapshot, it doesn't necessarily mean that all evidence of dynamic behavior is lost. If I were to ask you to try to order these people based on who jumped off the cliff first, I think, we're smart enough to know that, this person must have, or there must be some type of pseudotemporal ordering to these people. And likewise, we're all smart enough to know that gravity goes down. So given that assumption, we can actually provide a direction to the pseudotemporal ordering that, this person jumped off before this person, and it's not the other way around, but that assumes, of course, that gravity goes down. And even more so, even though we have this pseudotemporal ordering here, we can use the information embedded in the static snapshot to predict the future, let's say, positional state in this case of this person. Even though gravity is going down based on what we observe in terms of the spatial positions of everyone else, we can predict that at a future time point, this person will probably look a bit more like this person, as opposed to like being down here, even though gravity is going down. The information regarding the other people gives you some intuition, or allows you to infer some level of information regarding, in this case, the angular momentum at which people are jumping off the cliff. So this is really a similar intuition that we can take into our single cell transcriptomics data and spatially resolved transcriptomics data, where we can essentially leverage the continuum of transcriptional states of cells along developmental, dynamic processes to try to infer how these cell states may change over time. And in particular, we can leverage information regarding nuclear and cytoplasmic gene expression that we observe, in our single cell spatially resolved transcriptomic profiling data, and use that to predict the future transcriptional state of cells. So here we do have to make a couple of assumptions. So we assume from our basic Bio 101 that we can expect RNAs to be born in the nucleus and where they're transcribed. And we assume that transcription is happening at like a reasonably constant rate. But what we get is we can observe within each cell, what is the amount of nuclear mRNA for each RNA. And we assume that these RNAs are being exported out of the nucleus into the cytoplasm, where again, we can quantify, what is the cytoplasmic expression level for each RNA species in each cell. And eventually, these RNAs will go on to be made into proteins and then eventually be degraded. So given this very simple model of the RNA life cycle, we can essentially write a set of differential equations that captures the rate of change that we expect in the cytoplasmic RNA levels as a function of what's coming in minus what's going out. And what this means is that when we look across a population of cells, when we look at for example, if there is no change in the cytoplasmic mRNA level, this rate of change would be 0, and we would expect a certain steady state level in terms of the nuclear and cytoplasmic expression ratios. Whereas if a cell is actively upregulating a gene, then we would expect there to be more more active transcription, and therefore a higher accumulation of these particular gene species within the nucleus, and it would have a higher level of nuclear expression than what we would expect relative to steady state when we look across the population of cells. And likewise, for downregulation, if we observe less nuclear expression and higher cytoplasmic expression levels for this gene for the cell, then we would interpret it as being downregulated. And indeed, when we look at some of our real data, here each point is now a single cell, and they've been colored based on transcriptional state. But generally what you can see is indeed that there are some cells that have higher nuclear expression relative to cytoplasmic expression than others, and other cells with lower nuclear expression relative to cytoplasmic expression for this particular gene, MCM6. And otherwise, and likewise, for this different gene, KIF2C, this population of red cells that were upregulating MCM6 is now downregulating KIF2C, and vice versa for these other cell populations. So generally, it does seem like our real data is reasonably behaves as we expect. Or the underlying model reasonably captures what we see in the real data. And then, therefore, we can, for example, take the observed transcriptional state for an individual cell, where here, each column is a different gene. Red would be if the gene is highly expressed, and blue if lowly expressed in the current observed state for the cell. We can then, use these models to predict the future transcriptional state for each gene. So perhaps, for example, if we take this gene here, which is reasonably low in the observed state, so perhaps if the cell is here, it's currently not really expressing this gene very highly, but in the future we predict that it's at a very short future, small delta t, that is going to be upregulating this gene. So therefore we predict that the future transcriptional state for this gene and this cell is going to be a little bit higher, marked in red. And likewise, we can do that for every gene that we're able to fit these models for. So we can have this very high dimensional representation of the transcriptional state for the cell in its current observed state, and also the predicted future state. And in terms of visualizing this data, one approach that many in the single cell analysis field have been using is to do clustering analysis, because what we have is a large number of cells and a large number of genes. It's very hard to visualize this high dimensional matrix. So instead, we try to project it into a two-dimensional embedding, in this case using t-stochastic neighbor embedding. So here, each point is a single cell, and cells are closer to each other if they're transcriptionally more similar. And they've been further colored based on clustering analysis, which is supposed to capture cells of similar transcriptional state in this case. Again, this is actually the U2LS cells. So these are cultured cells where we don't necessarily expect many different cell types, but we do expect, for example, different cell states, possibly related to the cell cycle. And indeed, when we do differential expression among these different cell states, we're able to identify genes that are canonical markers of different cell cycle stages, like MCMs and the KIF2C and CCNF and so forth. And likewise, we can visualize the expression of these genes within the same embedding, and you can see that there are some cells that are upregulating this gene, MCM6, and other cells that are downregulating based on the current, or I guess it's expressing this gene versus not expressing this gene. And likewise, there are some cells that are expressing KIF2C and other cells that are not. So we can have this 2D representation of our high dimensional data. But beyond simply doing that, what RNA velocity analysis allows us to do is then project the future predicted transcriptional state onto this embedding. So in addition to, for each point here being a cell, we can also project its future transcriptional state, and visualize, the resulting connection with an arrow. So as you can see here, it seems like cells that are here their future transcriptional state is here, and for cells here, their future predicted transcriptional state is here, and it generally seems to kind of follow a circle. And perhaps unsurprisingly, based on some of the information I've already told you about how this is a cultured cell system and some of the genes I've highlighted related to the cell cycle, it's not surprising here that this is actually indeed reflecting the cell cycle. So we can, in this case, take the angular, I guess the angle as the pseudotemporal placement of these cells in the cell cycle, and essentially begin, rather than looking at t-SNE embeddings, we can now represent their ordering along the cycle as a pseudotime, and begin asking, what genes exhibit a variable expression as a function of pseudotime. Oops. Bit of a giveaway, but we can see, for example, here again, each point is a single cell, and they've been ordered on the x-axis based on their pseudotime, which is the angle and this in the circle. And y is an expression of different genes. So this is actually the MCM6 gene, where we can see it's being kind of upregulated in this group of cells, and it begins being downregulated. And alternatively, this gene is KIF2C, and we can see it's being, upregulated in this group of cells and downregulated here. And there's a pattern to this gene expression as a function of pseudotime. So we can actually then statistically test, what genes have, a significant proportion of their variants that can be explained by the pseudotemporal ordering, and in that manner identify up to 1,000 different genes that appear to be associated with the cell cycle. And then, we can also visualize this information in a number of ways. So here again, each point is a single cell, and they're being colored based on their expression of these different genes that we identify to be associated with the cell cycle. And you can begin just visually appreciating how, the cell really has to gradually upregulate a number of different factors in order, or I guess as it progresses through the cell cycle stages. So this approach, so if you're interested in learning more about this, I would recommend you check out the PNAS paper that I mentioned previously. And of course, even though I think as a proof of concept, I showed you how this approach can work in revealing, self-cycling in cultured U2OS cells, we can, of course, apply the same computational method to analyze cells within the developing brain. So this is actually cells of the developing mouse hippocampus, where, again, this is a t-stochastic neighbor embedding representation of the data. For each point as a cell, we measured the full transcriptome, what genes are being expressed, and we can try to represent that as a 2D embedding, where now, again, cells are closer to each other if they're transcriptionally more similar. And we can appreciate that there are certain cells that are more similar to each other and others that are more different than this, but similar to each other here. And based on our knowledge of various cell markers, we can begin annotating these different cell types as astrocytes and so forth. But then with velocity analysis, again, what it allows us to do is project a putative directionality to how these cells are related to each other in terms of their differentiation trajectories. And what we find is, in this particular example, even if you didn't know anything about neurodevelopment or neuromaturation, based on velocity analysis, you can tell that there are groups of cells here that seem to be the kind of starting origin that eventually leads to these cells, or alternatively, lead to these populations of cells. And indeed, these are the neuroprogenitors that give rise to the radioglia that might give rise to the astrocytes. And alternatively, this is the neuronal branch. So indeed, matches what we expect based on our known biology here. But perhaps more interestingly, I think with velocity analysis, in addition to providing a sense of directionality, we can also begin teasing apart, what genes are being actively upregulated at this differentiation fork that's going to be responsible for whether a cell, becomes a granule cell or becomes a CA cell and so forth. And I think so far, what I've shown you is that, with velocity analysis, we can project, these predicted future transcriptional states and the observed transcriptional states onto these 2D embeddings to visualize, these putative, differentiation trajectories and trends. But one thing you might have noticed is that so far, we've been projecting these velocities onto t-stochastic neighbor embedding plots. Which is only one possible, 2D dimensional representation of this data. We can also, use principal components analysis of like, PC1 or 2 to reduce this observed transcriptional state down to 2 dimensions, or diffusion mapping is another possible technique. And UMAP is a more recent graph-based embedding approach that's also similar to TISNE. We can use that to visualize our data in 2 dimensions. But as you see here, so here is an example of pancreas differentiation, where each point is again single cell, and they've been colored based on their putative cell types. And for pancreas development, what we expect to see is that there should be a set of cycling ductal cells that then give rise to endocrine cells and pre-endocrine progenitors that eventually will differentiate into the different hormone-secreting cells of the pancreas. But what you might be able to visually appreciate here is that depending on what type of 2D embedding we're using, you can kind of glean different insights or get different impressions of this true underlying kind of cellular trajectory. So for example, if we were to visualize the data with PCA, we really can't capture the cycling nature of the ductal cells. We do capture, for example, the differentiation of the ductal cells into the endocrine cells and the terminal hormone-secreting cells, but we also don't really see any branching of the terminal hormone-secreting cell types. So that type of, that granularity of information appears lost, at least in the first 2 principal components when we try to visualize our data this way. And alternatively, TISNE and UMAP might be able to better capture the differentiating into the different hormone-secreting cell types, but there are other potential limitations as well. So what Lila Atta, who's a MD PhD student on my team, sought out to do was rather than projecting RNA velocities into these 2D embeddings that were actually all established based on only the observed transcriptional states, to actually use the RNA velocity information itself, both the observed and the predicted future transcriptional state, to create a 2D embedding that is then able to create a more stable visualization of the underlying cellular trajectories. And she packaged it all up into a nice software that called VeloViz. And if you want to learn more about it, you can check out our preprint, which is on bioRxiv. But very briefly speaking, just to tell you a little bit about how VeloViz works. So again, we take advantage of the current and the projected, future transcriptional state of each cell. In this case, we might have 3 cells, A, B, and C. The solid circles are the observed transcriptional state, and in this case, the dashed red circle is the predicted future transcriptional state of cell A. So we have, that for all the cells. And what VeloViz does is compute a composite distance that tries to take into consideration both the transcriptional similarity of these predicted future transcriptional states to all other observed transcriptional states, as well as the directionality of that velocity. So in this case, the cell A has the predicted future transcriptional state is kind of going in this direction in a high dimensional representation. So this predicted future transcriptional state has a certain distance to the observed transcriptional state of B and C. And then there's also a velocity vector going from the observed to the predicted that is, I would argue, more so in the same direction from A to B than from A to C. So the composite distance tries to capture both of these aspects of the transcriptional similarity between the observed and the predicted future transcriptional states of all cells. And then we can choose, for example, then we can create a graph where each node is a single cell and an arrow will connect the nodes if, for example, in this case, the predicted, the future transcriptional state of A looks the most like B based on this composite distance. So we can create a graph where we connect, we create an edge between A and B. And we can do this, of course, for all the cells. And that allows us to create this very high dimensional graph, which we can then embed in 2D using various graph embedding approaches, such as force-directed layouts and so forth. But what this allows us to do is then, we can apply this approach to the pancreas development single-cell RNA sequencing example I showed previously. And, indeed recall that, we expect to see cycling ductal cells differentiate into endocrine progenitors and giving rise to the different hormone-secreting terminally differentiated cell types. And indeed here with when we construct our velocity-informed graph with VeloViz and visualize using a force-directed embedding, we can indeed, see these cycling ductal cells that differentiate into the endocrine progenitors and give rise to the different, beta, delta, epsilon cells of the terminally differentiated cell types of the pancreas. So it seems that this approach is able to reasonably faithfully recapitulate the underlying biology and capture the expected cellular trajectory. Okay. So far, what I've shown is a somewhat picturesque example where we actually have a reasonably uniform sampling of all cells along a developmental process. So that's to say, I have, for the friends jumping off this cliff, I have a pretty even distribution of, when they jumped off the cliff. So it's a good distribution or sampling of people along this dynamic process. But in reality, or I guess in some more challenging tissues, such as the brain, actually, we often may not get such a uniform sampling along a developmental process, either because of biological reasons. Let's say it turns out that cells are differentiating through this or progressing through this particular gap rather quickly, so we don't see them a lot in our single cell analyses, or alternatively, especially in the brain, certain cell types might be a little bit more sensitive to dissociation, so perhaps we might lose them in the data collection process. So we might not get this uniform sampling, and what this means for our directed pseudotemporal ordering is that approaches such as UMAP and TISNE and other dimensionality reduction approaches and graph-based embedding approaches might be able to capture, for example, that these three people are a part of the same dynamic process, and these two people are a part of the same dynamic process. But it's actually quite hard to link them to each other. So we can, simulate an example of this as a proof of concept, again, taking advantage of our pancreas single-cell RNA sequencing data, where now we can just artificially remove the pre-endocrine cells. So we've simulated a case of, what would happen if we just remove these pre-endocrine cells and again, try to visualize this single-cell data using and the corresponding velocities using the typical approaches. And what you see perhaps most strikingly, again, so this is data after we've removed the pre-endocrine progenitors, and now we run everything through PCA, TISNE, UMAP, or diffusion maps again on the observed transcriptional states and try to visualize the velocities. What you see perhaps most strikingly in the UMAP example is that the underlying cellular trajectory becomes a little bit disjoint. So you again have the cycling ductal cells going into the endocrine progenitors, but because the pre-endocrine cells are missing, there's no connection now to the terminally differentiated hormone-secreting cell types. So rather than capturing the underlying trajectory, it's a bit disjoint now, and if we didn't know anything about pancreas development, we might be led to believe that these cells are going off in this direction, and these cells are not at all related to the endocrine progenitors here, which would be an unfortunate interpretation of this result. So in contrast, if what we do in VeloViz is by taking into consideration the predicted future transcriptional state of these cells here, and we're able to actually connect them in our higher representational graph to the terminally differentiated hormone-secreting cells, and essentially bridge this gap. And what it allows us to do is then more faithfully recapitulate the true underlying cellular trajectory in spite of the missing intermediates, which are in this case the pre-endocrine cells. So this is the VeloViz embedding that was generated using the data with the missing pre-endocrine progenitors. And again, as you see, we can recapitulate the expected cycling ductal cell types that differentiate into the endocrine progenitors. And in this case, our graph is able to connect these two disjoint populations and visualize that in the 2D embedding to more faithfully capture the putative differentiation or eventual differentiation of the endocrine progenitors into some missing intermediate state into the 4 different hormone-secreting cell types of the pancreas. And I think even though this is I've shown a few examples using U2S cultured cells and using pancreas development and so forth, I think of course we can also apply these technologies and tools to tissues, including the brain. And here are just a few of the highlights of recent efforts in a number of labs to capture to profile at a single cell and often spatially resolved resolution the different cell types within different regions of the brain, like the preoptic region or motor cortex, the subventricular zone, olfactory bulb, cerebellum, and many more. This is again a bit of an older slide, so there have been a lot of newer developments in just the past year alone. So my group in particular were very excited to be applying these techniques and computational tools to study the developing mouse brain or developing mammalian brain, and trying to apply these techniques to explore different classes of progenitor cells and to potentially relate them to each other and identify these putative differentiation forks, and try to explore how these different cell states are organized within the brain. So so we're coming up on time. Just as a summary, I'd like hopefully I've been able to show you a little bit of how single cell and spatially resolved transcriptomics data they really demand new computational and statistical tools for us to be able to extract any biological insights. One potential limitation that I hope I should be able to show you is that data from these single cell and spatially resolved transcriptomic profiling technologies, at least currently, represent just these static snapshots in time. So so the cells are dead, and there's no way for us to follow them as they undergo dynamic changes. But we can use computational modeling with RNA velocity analysis to, for example, predict the future transcriptional state of the cells. And then furthermore, we can take into consideration these predicted future transcriptional states to create a more stable representation of the underlying cellular trajectories. And this approach is also available as a computational software called VeloViz that we've developed. And I think all of this will be a lot more exciting as we as we begin applying these technologies and analyses to more complex tissues such as the brain. And with that, if you're interested in learning how to do some of these analyses, you're welcome to check out our lab website at jeff.works. We have lots of tutorials and sample data for you to download, so you can explore how to do these analyses yourself. And I guess also reproduce some of the figures that we have in our papers if that's of interest to you. And with that, yeah, thanks everyone for your attention. Again, I'd like to thank my team. This is our lab holiday photo, which was virtual this year, but none of this would have been possible without them. And with that, I'd also like to highlight my postdoctoral mentor, Xiaowei Zhuang, Chenglong Xia, who led the MERFISH effort on U2S cells, along with George Emanuel, as well as Guiping Wang, who led the effort on MERFISH in neurons, and likewise my PhD lab mentor, Peter Kurchenko, and Ruslan Soldatov, a postdoc in the lab, who worked with us on the velocity analysis. And with that, you're always welcome to find us online at our lab website, as I mentioned. Also feel free to reach out via email, and once in a while I can be found on Twitter, so definitely feel free to find me there too. And with that, I will stop sharing my screen so I can see you all.
Jean, thanks so much for joining us today and for your very interesting talk. I'm so sorry I was a few minutes late. I missed my chance to introduce you. So while it's a bit unorthodox, I hope I could give you a bit of a post-hoc introduction before we open the floor to questions.
Sure.
Dr. Jean Fan received her undergraduate degree in biomedical engineering and applied math at Johns Hopkins. She did her PhD in biomedical informatics at Harvard Medical School, working with Peter Kurchenko and Catherine Wu, where she developed computational and statistical methods for characterizing single-cell heterogeneity. She did her postdoctoral training at Harvard with Dr. Xiaowei Zhuang in the Department of Chemistry and Chemical Biology. And recently she moved back to Johns Hopkins as an assistant professor in biomedical engineering, where she's also affiliated with the Center for Computational Biology, the Center for Computational Medicine, and the Department of Computer Science. So as you've seen, her work today focuses on developing machine learning and statistical methods for studying multi-omic sequencing and imaging data. In addition to her academic accomplishments, Jean was recently listed as one of the Forbes 30 under 30 in healthcare. And she started a nonprofit company called Customized, which makes personalized STEM picture storybooks for young girls. I think this is such a great contribution toward promoting diversity in STEM. And I've already got one for my daughter. This is her diploma as an MD, which she's going to be in 25 years. So over the past few months, we've had the opportunity for many interesting discussions about her work with MERFISH and the mouse brain, which is why I suggested inviting her to give this seminar. I think this interface between imaging and genetics is so fascinating and is a chance to bring together some different communities of scientists, particularly people who are working with macroscopic brain imaging and people who are focusing more at the cellular level. So with that, I'll open the floor to any other questions. And if no one wants to volunteer to go first, I can start with the first question. So Jean, how large of systems can you apply these types of techniques to? And what kind of challenges arise when you're scaling these things up?
Yeah, it's a great question. So some of the images I showed within tissues, for example, we're still quite limited to millimeter scales of tissues. And the reason for that is largely a limitation of imaging time. So in order to do MERFISH, we essentially have to flow on the hybridization, hybridize, the various oligonucleotide probes, image, and then, image across a Z stack, and then move to the next plot, image across the Z stack, move across the next plot. We might do, 100, 200 rounds of imaging, and then, you wash off the the first bit, and you hybridize the second one, and repeat that imaging. So it can get, in terms of, the amount of data we generate, it can be quite large, in the terabytes for just a very small, let's say, 6 millimeter, 6 by 6 millimeter type region. So if you were to try to, scale this up to a whole brain, it would be, imaging time alone would be quite challenging. So you would probably want to parallelize by, region or across many microscopes. And yeah, otherwise it's just going to take too long. But currently we're still in kind of the millimeter centimeter type scales for the size of a tissue that we can image, at least in a single experiment.
Thanks for your answer. So we have another question in the chat from Zhuking Wang. How do you know if there are missing, oh, it's scrolling away. How do you know if there are missing cell intermediates? Zhuking, could you expand on that a little bit? I'm not sure if it's totally clear from the text.
I think I understand the question. So just allow me to paraphrase. In the second part of the talk, where I was discussing how to take into consideration RNA velocity and creating the 2D embeddings, representations of the cellular trajectories, I showed how, if there are missing intermediates, like a gap in the underlying, cellular trajectory, then VeloViz is, by taking into consideration the future predicted transcriptional state, you can kind of cross that gap and still recover the underlying cellular trajectory. So the question is then, how do we know if there is a gap? Of course, I show that if there is a gap, then this method is better, but how do we know? And I think it's a very good question, and I think one way that we have, for example, in certain contexts, we expect there to be a missing, we expect there to be missing cell intermediates because we biologically know that this process occurs rather quickly. So we often only see cells at the beginning of the trajectory and at the end, and we don't see that kind of intermediate transition. We can also, for example, in other more cancer-associated applications that we're taking a part of, where we have, let's say, single-cell sequencing and spatially resolved MERFISH, of the same tissues, we can see that there are certain cell type proportions are very different. Even though we're assaying the same tissue, and the one possibility is that in dissociating the cells for RNA sequencing, there are certain cell types that are going to be a little more sensitive to the dissociation process, and they might lyse, they die. So then we see a fewer of these cells than what we would expect if we don't dissociate. So that, in theory, could lead to missing cells, and those missing cells could be of an intermediate developmental state, for example. But generally, I think I would encourage people to try out a couple different visualization approaches. And if it does seem like there are that you're getting somewhat conflicting or different representations or visualizations, as I showed, with the UMAP, you get 2 distinct clusters. With VeloViz, you get a continuum. If something like that happens, that could be suggestive that there might be some missing intermediate cell types. But generally, yeah, it's a great question. It's a challenging one.
Thanks, Jean. The next question is from Farid Rashidi. Is there an attempt to check RNA velocity on tumor samples? Do the assumptions that you've made hold there?
Yeah, really excellent question. So for RNA velocity, I guess there's two RNA velocities. One is RNA velocity, the original, which was developed in my PhD lab in Peter Kurchenko with Stella Narson's group. And that approach is based on spliced versus unspliced RNA. So looking at the ratio of the spliced versus unspliced under the assumption that genes are, first transcribed in an unspliced format, they're then spliced and turned into proteins and degraded. So one concern that I've always had with velocity analysis when we look at spliced and unspliced as applied to tumor samples is that a lot of tumor samples have dysregulation in alternative splicing. So that could lead to, for example, aberrant intron retention. So then, your quantification of the unspliced RNAs, it would be less of a reflection of, nascent transcription and more a reflection of this, aberrant process. And so in theory, that could lead to some issues with your estimations of RNA velocity. And I guess in theory, the other RNA velocity method that, I said that there were two. The other version is what we call RNA velocity in situ, which is based on the nuclear versus cytoplasmic expression quantification. In theory, I guess that should be more robust to the aberrant alternative splicing, but it's purely feasible or possible that tumor samples could have aberrant nuclear retention, so that the nuclear expression levels you observe are again not the reflection of nascent transcription, but rather an aberrant retention of the gene within the nucleus. So then you would again have this issue with not necessarily holding true to the underlying assumptions of RNA velocity. I think I have, so those are, it's generally, to get back to your question, those are, I think, some of the challenges with velocity analysis in tumor samples that, we should be aware of. I haven't seen yet any kind of systematic attempts to evaluate whether velocity analysis works, for example, in tumors that have known splicing aberrations. But it's also not clear to me that, I guess where it would cause an issue is if it impacts, a large number of gene species. If it only impacts one or two, it probably, won't impact the overall velocity estimations, or the, predicted future transcriptional state for the cell as a whole won't be affected, but it might affect one or two genes. So first, to really have an impact, it needs to impact lots of different genes. And two, I think it would need to impact a subpopulation of tumor cells in a very specific way, a coordinated way, that we would mistake it for upregulation or, differentiation or, moving in a certain, towards a certain transcriptional state. So it would need to be a very coordinated type of, confounding in terms of, the intron retention or nuclear retention. But I think it'd be really interesting if somebody looked into how well these velocity estimations, one, were different in tumor samples compared to matched normal, and likewise, are these differences attributable to systematic intron retention or nuclear retention?
The last question we have in the chat is from Krista Caggiano. She says, do you think multi-omics approaches like those that profile RNA and methylation would help in the pseudo-time assembly?
Oh, yeah, that's really interesting. I wonder, it's a very interesting question. I guess, I've been thinking about how we can try to leverage, for example, let's say, single cell attack sequencing, single cell methylation profiling, and whether that would reveal anything in terms of pseudotime assembly. The only thing I can, really think of off the top of my head is, if we were to construct the underlying cellular trajectories based on the RNA information, and then see, that there's a very smooth, gradual upregulation of smooth transition of methylated states, or let's say a gradual increased accessibility of certain sites. Although I guess in single cells it's rather binary. You're either methylated or not, or accessible or not. Whereas RNA, there's much more dynamic range. So in that sense, yeah, it's hard for me to see how we would integrate that information directly in the construction of the pseudotime itself, and rather more like a kind of a secondhand after we've created the pseudotime from RNA to try to, I guess, validate that based on the patterns of methylation and accessibility or something like that. But perhaps, but yeah, I think it's a really interesting direction. Perhaps there's smarter ways of aggregating across multiple genes or within a pathway or something, or multiple promoters to try to get at a more dynamic range or something like that. That could be useful. But yeah, it's a very interesting question.
Okay, thanks for your answer. If there's any other questions, you can type them in the chat or just feel free to speak up before we finish. If not, then let me just thank Jean again. Turn the host back to Shantanu.
Yeah, thanks, Daniel, for the nice summary of the talk, and thanks, Jean, for a really beautiful talk. I just have one last question. You, in the TSNE plots, you had a circulation of the vector fields. Do you think is that an artifact or that's expected?
Oh yeah, in that particular case, it is expected. So these are cultured osteosarcoma cell line cells, where we don't really expect them to be. It's not like in the brain where we have many different cell types. They're all one cell type. The only transcriptional heterogeneity we expect to see from them is that they're cycling and dividing. That's kind of all they do. So it was nice to see that we were able to identify these transcriptionally distinct clusters that appear to upregulate different genes that we know to be associated with the cell cycle. And then what the vector field then was able to show is how these different transcriptional states were connected to each other. In this case, it just so happened to progress in a circle. And indeed, when we interpret, what genes are being upregulated, we can see that, the cells upregulating, G1S genes are preceding the cells upregulating the, S phase genes are preceding the cells upregulating the M phase genes in the vector field. So we definitely do believe that it is true. And furthermore, yeah, if you're interested, I would encourage you to check out the paper where we were then able to identify a lot more cell cycle related genes using the ordering of cells along the vector field.
That's interesting. Well, okay, so let's thank Jean again for such a nice job. And she's available over email if any of you have any questions. And thanks again, Daniel, for introducing us to Jean and bringing her here. So this concludes the seminar. See you next month. Bye.
Great. Thanks, everyone.